This is Goddess of Science Biology, and I'm Mrs. Bolvin, and we're going to do part 12 of our cell structure and function series, and this one is about active transport. Okay, so just to recall from previous lessons, passive transport does not require energy by the cell to move molecules around. Active transport is quite different in that it does actually use energy to move molecules across the plasma membrane. Sometimes it's moving larger molecules either in or out of the cell. Sometimes it's just moving things against the high concentration to low concentration gradient that usually happens. The high concentration to low concentration happens automatically, just random movement of molecules, but to move something against that, it takes some energy by the cell to do that. So usually moving against a concentration gradient unless you're talking about large molecules leaving or entering the cell. So active transport, when it's moving against the gradient, moves from low concentration to high concentration. And the first one that we're going to talk about is something called a sodium-potassium pump. Now the reason that there's these little plus signs here is because they're ions, which means they're an atom with a charge. Okay, no big shakes there, just keep in mind that they have a little bit of a charge to them. So the way that a sodium-potassium pump works is it needs ATP, which is a huge energy molecule. All living things use ATP, the same ATP, to fuel these kinds of activities, to fuel just about everything that happens in the cell. So what happens is on one side of when the pump is open to the inside, sodium and ATP on the inside binds to the pump. When the ATP splits apart, that changes the shape of the pump and then it opens the pump to outside. So the sodium can go away. And this pump is made out of protein. Anytime you're binding something to a protein or you're taking something off, unbinding it, if you will, from a protein, it's going to change shape. So once that sodium goes away, some potassium can go in there on the other side, and that's going to cause the pump to change shape again. And so then it reconfigures itself so it's open to the inside. Okay, so I'll have some videos for you to watch that kind of explains this a little bit better with some visuals for you. The second type of active transport we're going to talk about is endocytosis. The word endo, you can kind of think about this as in, and then cytosis, that's like cell eating, I guess. Okay, so what endocytosis is it uses energy to bring large particles into the cell. So it's kind of an interesting thing that happens here. A particle touches the membrane surrounds it, and then draws it inside with the membrane still around it. So it makes like this little pocket, which is kind of interesting. So this little piece of whatever, it could be food, it could be a bacteria, it could be a virus, could be, anything. Once it touches the membrane in a particular place, it will cause the membrane to form around it. And then eventually the two sides of the membrane here will pinch off and make this little membrane-bound organelle called a vacuole or vesicle. And then it waits for when the cell is ready to break this down, and it just kind of makes its own little storage container. So there are two types of endocytosis depending upon what you're moving inside the cell. There's phagocytosis, which is solids doing the whole endocytosis thing, Or there's pinocytosis, which is liquids doing that, large liquid molecules. The way I remember it is pino has an I in it, second letter, and so does liquids. If we have a process for large particles coming into the cell, we must also have a process for large particles moving out of the cell. That's exocytosis. Exocytosis is using energy to move large particles out of the cell. And it, as you might imagine, is kind of the reverse of endocytosis. So remember back when we were talking about the organelles and what they do, and we said that the Golgi apparatus took whatever the endoplasmic reticulum produced and packages it in a vesicle. And what happens to those vesicles packaged by the Golgi apparatus is what happens is part of exocytosis, excuse me. So those little vesicles packaged by the Golgi apparatus moves to the membrane, fuses with the membrane, and then the particles get expelled. So we have, here's our vesicle made by the Golgi apparatus. It goes to the, and since it's surrounded by membrane material here, it's no big deal to fuse with the plasma membrane. And then once it does that, it just becomes incorporated into that membrane that was surrounding the vesicle, becomes part of the plasma membrane, and then whatever was inside the vesicle now can be on the outside. Well, that's it for this time. Thanks for joining me. Have a great day.